target identification, the target should be the molecule which is present within the organism. That should be in the organism. If you are working on the human body, the molecule should be present in the organism. What are the approaches that from where you can get the target? Literature mining is one of them. Protein expression, structure function studies, the pathway study can also be done, database mining, and high throughput screening. All these methodology can be used to identify the target for which, against which you want to design the drug. Okay. While considering the drug, make sure that it should be relevant to the physiology of the disease. It is not like, okay, you take any XYZ target and it is not playing very important role in the cause of or pathogenicity of the disease. That should be very much clear. So after identifying the target, we have to validate it, okay? And during the validation, we have to answer few questions. It should affect an appropriate biological response, okay? Like it should not be like whatever the compound, whatever the target you are considering, in the absence of that target, if any other thing is doing the same thing, then there is no meaning of considering that target. You will say that particular given target is causing the important role. It is responsible for the pathogenicity as well. Okay, but in the absence of that particular target, some other protein or some other gene is doing the same thing, then there is no use of that. Okay, so this targeted gene disruption terms which we further use to target validation. That means virtually we do this, that if we disrupt this target, what is happening? Is the procedure same? If the procedure is same, we will not consider that target. If the procedure is affected, we will consider the target. Okay. Then comes the lead identification. So this lead identification, like whatever we have to identify the lead compound that must exceed a specific potency. Okay, what is the potency threshold against the target? That means it should bind to the active site. It should remain bound to the protein under the external conditions as well. All these. Okay. These leads can be from any sources. Either you can synthesize them, you can get it from the molecular libraries as well. Okay. And the techniques that can be used are virtual screening, chemoinformatics, chemical genetics, all these are the things that can be done. Okay. What is, okay, leads has been identified. How will you validate them? Okay, once the lead compound is identified, its states need to be optimized. That means, if you remember, I told you the compound which you are considering, whether it is safe or not. If the compound is good, well fit to the protein, everything is good, but it is not safe for the human body, it is not safe for the organism you are working on, then you can't consider that. Okay. Once now here we are having the target, we are having the ligand. We are having the ligand. Now we'll go for the docking. Docking is what? That here we are basically positioning the ligand into the active site of the protein or the target. Okay. While binding, this positioning of the ligand into the active site of the protein, while this thing is happening, some energy will be released. Okay, so what are we have to calculate the binding affinity. So it is not a process, okay, the protein and ligand are binding, that's it. No, we have to consider many more things. The lesser energy should be used there. Okay, because whatever the energy it is acquiring, that will be used from your body. Now, if you will, they will bind together. Okay, so the lesser energy should be used. Okay, so these docking algorithms which we use to find the ligand, the confirmation of the, basically whatever the experimental structure you will be having, what you have to check at minimum binding affinity, which ligand is binding to the structure or the protein. Okay, it can be multiple proteins, single ligand, it can be single protein, multiple ligand. Both things are possible. Okay, there are many tools. Few of them I'm naming here: the Autodoc, Bina, Gold, Glide, Hex. These are the tools. Gold and Glide are the, like, they need to be purchased, but Autodoc and Hex are freely available. Okay, that is the mechanism of action. This is what your protein is, and that is what your drug is. Okay, that is before docking, and after docking, it will come and it will just close it, and the drug will bound somewhere in the protein, and that wherever it is binding, that should be the active site of the protein. Only then the drug will work, otherwise it will not work.